All right, we start out talking about water because from our handout, one of the major 7 themes in our course is that all cells, every cell of any kind, plants, animals, bacteria on this planet, whether they live in the desert or in Antarctica or in your body, all cells exist in a water environment. And also water chemistry is a great way to introduce a lot of chemistry. So we'll talk about water, and we can imagine a cup full of water, and that cup can have stuff dissolved in it, so we can just zoom in on the water in the cup, and we can see this. Maybe there's some sugar molecules dissolved in there. Maybe there's a bacteria in there. Biologists always talk about concentration of molecules because it's always concentration that matters. The bacteria doesn't care how many total molecules of sugar are dissolved in the water. It cares about their concentration, because the concentration is what determines how frequently the bacteria will bump into sugar and get to eat. So at this concentration, the bacteria will eat at a certain frequency. If we double the concentration of sugar, the bacteria would eat twice as often. And that's just the key concept, and it's true whether it's bacteria in a cup of sugar water bumping into food, or whether it's your body, and in your body a hormone in the blood bumping into a receptor. It's always about concentration, and concentration is the number of, say, sugar molecules in a volume of liquid. In biology, we're almost always talking about stuff dissolved in water. So numbers of sugar molecules dissolved in a volume of water. If we double the water, the concentration of sugar molecules is cut in half. If we double the sugar molecules, the concentration of sugar molecules is double. There's so many molecules dissolved in biological systems that we, instead of using numbers, you might say something like, oh, well, how many dozens of sugar molecules are dissolved in that cup of water? There are even more than that, and so the convenient number that we use is not a dozen equal 12, but rather a mole. And a mole is just like a dozen, except for it's a really big number. And biologists talk about how many moles of molecules are dissolved in a liter. The unit for that is molarity, and molarity is big M, and big M equals moles per liter. In a mole, there are this many molecules, and the molecules in biology are really small, so we have this, that there are that many molecules of water in just a sip of water. So it's a huge number of molecules that we're talking about that makes up 1 mole, yet they fit into a really small volume. This is what water looks like. It's H2O, so we know about that's the structure of the water molecule, and it's made up of 2 hydrogens, those are the circles that are white, and then 1 oxygen, that's the circle that is red. So that's the structure of water molecule, and they're dancing around, floating around, and make up a solution of water. Water molecules, if you look inside of them, have a really significant structure, and that is that in the electrons that they share, which we'll talk about in class, the oxygen is got more attraction to the electrons than the hydrogens, and as a result, there's a little bit of a positive charge near the hydrogens and a little bit of a negative charge near the oxygen. As a result, water molecules can be attracted to each other. So in a solution of water, this interaction is occurring, and these are really unstable interactions, so they're being made and formed. One way I always talk about this is, imagine that a water molecule is like a married couple, and when they're in isolation, say at home, they have a really good marriage. But if they go out dancing, each of the partners is going to be attracted as they dance and swirl around the dance floor at a crazy dance party. They're going to be attracted to other water molecules, and that happens. And so really a cup of water sitting on your table is really a crazy dance party where these water molecules are dancing around, and each water molecule is a stable married couple, but each one has attractions to other water molecules like that. And that's happening, that crazy dance party is happening, whether the water molecule or whether the water is sitting on your table in the day or at night. So just all the time, water is dancing around like that. And here's something can happen. Basically, the water molecules can get divorced and remarried. Because of this attraction, the hydrogen from one water molecule can dance away and join another water molecule. What's happening here is a chemical reaction, and so that's shown here. So it's a bit of chemistry. Chemical reaction is where chemical bonds get rearranged, and that's what's happening here. So it's a chemical reaction. And we'll talk a lot about chemical reactions as we go through our course as our first one. And that's happening because although we're just drawing these two water molecules, there are other water molecules dancing around these guys, and they're destabilizing these bonds. So this hydrogen goes dancing off because this oxygen got attracted over here, like that. It really is a crazy dance party. And when this hydrogen goes away, this oxygen will grab a hydrogen from another water molecule. And of course, at that time, this oxygen, who's already attracted to some guy over here, will grab him and start dancing with him. And there's this chain reaction. Like, for example, here, this hydrogen is not very strongly attracted here, and this hydrogen will go dancing off with a water molecule that's just lost her husband over here, like that. So there's this chain always going on through water, just this kind of dancing chain of lightning going through the water of partners changing, and that's a really important part of water chemistry. If we could sort of take a snapshot, sort of take a photo, we'd see something like this in water, but it would just be vanishing because it's so shifting around. Instead of imagining that image, what biologists always imagine is this image. They imagine that on the way from here to there, you could take a snapshot and see this, just for a split second. And we'll talk all the time in this course, because biologists always talk about this, that to represent these breakups and reunions, we talk about the concentration of this hydrogen ion. It's a hydrogen ion because it lost an electron, and it's going to gain an electron. And in between, it's got a positive charge. Over here, an extra electron gives this a negative charge. And so we'll focus on this guy, the hydrogen ion. And anytime you see this in biology or chemistry, that stands for concentration. Those square brackets mean concentration. So here we're talking about taking a snapshot of the water in order to count the number or the moles of hydrogen ions per liter, or the molarity. So we're talking here about the hydrogen ion concentration molarity in water. And that can change. We can have higher or lower levels of it. And when it changes water chemistry. So we want to keep track of that, and scientists do by a unit called pH. And pH is equal to the negative log of the hydrogen ion concentration. And we need to describe what that means. So to start out with, see that at an acidic solution, you have high numbers, high concentration of hydrogen ions. Then at a neutral solution, the neutral pH of 7, the hydrogen and hydroxide ion concentrations are the same. And then we keep going down with hydrogen ion concentrations until we have this ratio with a lot more hydroxide ions than hydrogen ions. You can think of these as like single women and single men. And anytime you inject that into a dance party, the more you inject, the crazier it's going to be. So at a neutral pH, things are going along, everybody's dancing, but just having pretty good fun. And but as we go to these extremes, the water chemistry really changes. It's the negative log of the hydrogen ion concentration. And what that means is that at a pH of 4, the hydrogen ion concentration is equal to 10 to the minus 4th molar, because it's the negative log. So we take the negative log of this, and that is equal to pH 4. Something that's significant about that is that when we go from one pH unit from 4 to 5, we're going to be decreasing the hydrogen ion concentration by 10 times, because we go from 10 to the minus 4th down to 10 to the minus 5th. So one pH unit change can have a dramatic difference in change in the water chemistry. That change in the water chemistry affects the stuff that's dissolved in it, and life exists in water. pH of water is really significant. Here's from your book also, just kind of an example of how the shift in pH can really affect life. So that's the message for that. And we're going to go back to this cup again for talking about bacteria and sugar dissolved in it, because biologists are, instead of talking about the molarity of water, they're almost always talking about the molarity of molecules dissolved in water. So back to this sugar and the idea of sugar having a molarity, a concentration in water. And here we're looking at two kinds of sugar. We have one sugar called glucose and another sugar called sucrose. It turns out if we looked at the molecules, sucrose is about twice as big as glucose. And so I've drawn it this way. It's important to look at this, though, that the molarity of glucose in this cup right now is equal to the molarity of sucrose. So the frequency that the bacteria would bump into glucose is the same as the frequency that the bacteria would bump into glucose. They're both the same. And that's significant in biology, again, back to that. So we want to be able to measure that. We also want to be able to create solutions that have that. So for example, if we had a cup of water and we wanted to add sugar to it, in order to make a 1 molar solution of glucose, we'd need to take 180 grams of sucrose and add it to water, dissolve it in water, until we had 1 liter of water. And then we'd have 1 molar glucose. If we wanted to do that same thing, make a 1 molar solution of sucrose, see, we have to add more grams. And that's one of the main reasons for defining the molar unit, is to be able to make solutions. In the case of sucrose, we'd have to add 342 grams of sucrose and then bring it up to 1 liter to get a 1 molar solution of sucrose. And we'll have questions like that. You know, how would you make a solution of this or that once we've given you the molecular weight or the mass, gram molecular weight of a molecule. They are different. Each one's different. And this is just some of those terms defined that we already talked through in this handout. So it's written out as sort of a summary. The mole is the specific number. Then molecular weight is what I just talked about. Each type of molecule has a distinctive molecular weight that we can calculate. For example, the molecular weight of water is 18. The molecular weight of sucrose is 342. The molecular weight of glucose is 180. And we'll learn how to calculate that in class, but typically that's something that would be given. The next thing is this gram molecular weight. The idea that since sucrose weighs 342 grams, if you weighed out 342 grams, you'd have 1 mole of sucrose, like that. So there's a correspondence between molecular weight and gram molecular weight in order to calculate numbers of moles. And the final thing is the concentration, molarity, which is the unit moles per liter. And that's what we started with. So 1 mole per liter is a 1 molar solution. And then here's just this idea that if you had a 3 molar solution, that'd be 3 moles per liter. And to make a 3 molar solution, you would take 3 moles of glucose and dissolve it into a 1 liter of up to 1 liter of water. And 3 moles of glucose would be this many grams, because 1 mole weighs 180 grams and we want 3. And that's it.